Okay, so now we continue with our lecture on motility. So if you remember, we started discussing motility in the oral cavity. We discussed those processes of swallowing and deglutition and how the bolus is going to move from the oral cavity into the pharynx, from the pharynx into the esophagus, all the way into the stomach by opening of the upper esophageal sphincter and the lower esophageal sphincter. Then once the bolus moves into the stomach is involved in mechanical contraction that will bring about mechanical breaking down of the bolus, mixing it with gastric secretions. Then it will initiate the digestion of proteins, especially because in the stomach, we said that's where you find a lot of pepsin, and pepsin is produced by the chief cells as pepsinogen. But once it has been converted into pepsin, it will initiate the digestion of proteins in the stomach. Then there's also a bit of gastric lipase that will also proceed with the digestion of fat, but that digestion is not complete in the stomach. It's just partial digestion because you don't have a lot of gastric lipase. Then there are mixing contractions taking place within the stomach, which are some kind of peristaltic contractions. Then we also have emptying of the gastric chyme or emptying of the gastric. So gastric emptying is controlled by some hormones and also the nerve that is intrinsic to the GIT. So you have the plexuses, remember, the submucosa and the mesenteric plexus that are involved also in gastric emptying. And once the chyme now moves into duodenum, so there's emptying of chyme into the duodenum, there it will trigger certain motility again. So this is where we are starting from. Motility in the small intestines. So the mechanism that we are going to discuss here, which are called peristaltic contractions or mechanism, you need to understand it very well. It can make a very good question for an essay for you to explain the mechanism of peristaltic contractions. Okay, so we start now. So let's start by looking at the function of intestinal motility. What is the function of intestinal motility? Why do you want to have motility in the small intestines, in the large intestines? So what would be the function? So I'm sure you can guess by now that one of the functions of intestinal motility is to mix the chyme with digestive secretions. So you need to mix the chyme with the digestive secretions because in the intestines, you have intestinal secretions. Some of them, they're coming from the accessory organs like the gallbladder or the liver and also the pancreas that will contain bicarbonates and also enzymes. So these intestinal motility, they will mix the chyme with the digestive secretions so that digestion can proceed within the small intestines. So you need to mix it with the enzymes for digestion to take place and also for absorption to take place. So one of the functions is to mix the chyme with the digestive secretions, then to bring the fresh chyme into contact with the absorptive surfaces of the microvilli. So this is going to facilitate absorption. So as you are mixing the chyme, you are also spreading the chyme on the surfaces that are involved in absorption. So that motility can also facilitate absorption in the intestines. Then the other function is to propel the chyme toward the colon. So whatever chyme that is not digested, it's not going to be absorbed. So it has to move towards the colon for the production of the feces or fecal material. So it will end up into the colon, and then later on it will be excreted via the anus. So whatever is not being digested, it has to move. So you find that it is the function to propel this chyme towards the colon if it's not digested. So this is more of anatomy, and we already discussed the functional anatomy of the GIT, of which you know to say the first part of the small intestines is the duodenum, then followed by the jejunum, then the last part is the ilium. So the portion between the small intestine and the ilium, you have a sphincter that is called the ileocecal sphincter. So you can see here the ileocecal sphincter or the ileocecal valve that is separating the large intestine and the small intestines. But now we need to appreciate the motility that is taking place within the small intestines. So types of movements. What are the major types of movements that you can appreciate in the small intestines? So there are rhythmic segmentation contractions, which are also referred to as segmental contractions or mixing contractions. So these are called rhythmic segmentation contraction or segmental contraction or mixing contractions. So you have certain segments of the small intestines that are contracting and other segments are relaxing. Then at a next point, maybe you find that others are contracting, others are relaxing. So they are just exchanging. So certain portions are contracting, other portions are relaxing. So that will bring about mixing contractions. They are not there to propel the chyme from point A to point B. They are just there to chop the chyme or to mix the chyme. So they are called mixing contractions. Then we have the pendular movements. So the pendular movements, these are swaying movements of the intestines. So swaying side by side movements of the intestines. So they are called pendular movements. We are going to discuss in detail for each type of movement. Then we have peristalsis. So there are peristaltic mechanism that will help now to propel the chyme from point A to point B. Those are called peristaltic contractions. Just like you saw in the esophagus, to say when you swallow the bolus, it's going to be pushed down to the stomach via peristaltic contractions. Of course, gravity has got an effect on it, because if you are swallowing a bolus which is heavy, gravitational force will also facilitate it to move very fast to the stomach. But you know to say that the smooth muscles or the esophagus, they can also contract to push the bolus down the esophagus. That's why even if you swallow when you're lying down, the bolus will still move. Even if they tie you upside down, the bolus can still move towards the stomach because of the peristaltic contractions. Then you also have movements of the villi. So you have the finger-like projections, which are called villi. Then on those finger-like projections, you also have tiny projections, which are called microvilli. That's where you find brush border enzymes that will finish up the digestion and also to facilitate absorption. So those villi, they can also move in a particular manner. They are those more like beating movement or lashing movements of the microvilli and the villi. That is all because we have the muscularis mucosa associated with those villi. So they are able now to move as well. They have a bit of motility as well. That will facilitate absorption and digestion as well. Okay. So let's start by discussing segmentation contractions. Segmentation contractions. So a loop of intestine is divided into a number of segments of nearly equal size. So there are tiny segments. So a loop of intestine, they are divided into those segmentations or segments. In the large intestines, they are called house striations or haustria. So those segments, they can contract, they can relax to bring about segmentation contraction. So as one set of contraction relaxes, a new set begins, but the contraction occurs at a new point between the previous contractions. So this will facilitate the chopping of the chyme or mixing of the chyme with the secretions. So it helps to chop the chyme 2 to 3 times per minute. So these segmentation contractions, they will help to chop the chyme 2 to 3 times per minute. So they are simply just mixing the chyme. So I hope you are also able to see what I'm projecting. That is an illustration of segmental contractions. So you can see certain segments that are contracting, and then other segments are relaxing. So those segments that we are contracting previously, they are going to relax. Those that we are relaxing, they are going to contract. So they are just more like random contractions and relaxation on the loop of the small intestines that will facilitate mixing and also chopping of the chyme. So these kind of contractions, they are not interested to propel the chyme or movement of the chyme from point A to point B. They are just mixing and also chopping the chyme. Okay, so these are called segmentation contraction. There are segments contracting, other segments relaxing. Then they will exchange just like that. Okay, so we proceed. How are the segmental contractions controlled? So the control of segmental contractions. So of course, we have the interstitial cells of Kagel that are involved to control the rhythm of motility. Remember, I told you to say the interstitial cells of Kagel, they will set a rhythm at which the smooth muscle cells are going to contract. Why? It's because they have special types of channels, the slow calcium sodium channels, which will bring about the slow waves, the initial depolarization. So that initial depolarization, if it depolarizes to a threshold, then they will fire spikes, which are called spike potentials. And the spike potentials are the ones that will stimulate smooth muscle contraction. So especially the mesenteric plexus, that is between the longitudinal muscle and the secular muscle, they're the ones that are going to be stimulated by the by the interstitial cells of Kagel. So you find that these smooth muscle cells, they are going to be stimulated and then they'll contract, of which we said that the muscularis externa, composed of the secular muscles and longitudinal muscles, are the ones that are involved in motility or the GIT. Then the muscularis mucosa, they are mainly interested in absorption and also the movement of the microvilli, like I explained. So for you to have the segmental contractions, you need the longitudinal muscles and the secular muscles to contract. So that is being controlled by interstitial cells of Kagel. Okay, so I hope you still remember the general control, the GIT, that lecture that we had. I explained everything. So you just need to remember that. What about the frequency of segmental contractions? So the frequency of segmental contraction is directly proportional to the frequency of slow waves. So it's dependent on the slow waves. The more slow waves you have, the more the spike potentials, the more the motility. So the frequency of segmental contractions is going to increase once you have an increase in the slow waves. Then the strength of contraction is direct proportional to the spikes. So the spikes are the actual action potentials that will bring about contraction. So the force of contraction or the strength of contraction is dependent on the spikes, because the spikes are the ones that will produce the actual contraction. They are stimulatory to the smooth muscles. For the smooth muscles to undergo depolarization, then to contract. So this frequency is controlled by the amplitude of the slow waves. So the amplitude of the slow waves, if it's increasing, you will see a lot of spike potentials, you will see a strong contraction, and the frequency of segmental contractions is also going to increase. So on top of this intrinsic nervous supply to the GIT that is controlling the segmental contractions, you need to understand that there is a hormonal control of GIT motility. So the motility of gastrointestinal tract system is also affected by specific hormones that are produced by the GIT itself, or they are produced by other glandular cells within the body. So down here, you have some of the hormones that are stimulatory to the segmental contractions. So you can see the first one there is gastrin hormone, of which we said most of this gastrin hormone is produced in the antrum of the stomach. So the antrum of the stomach, they do contain the G cells, then a bit of the duodenum. The duodenum can also have a bit of G cells that are producing gastrin. This gastrin is a hormone that will bring about depolarization of the interstitial cells of Kagel and also depolarization of smooth muscle cells. And that will cause motility, because you know to say, once the cells are undergoing depolarization, they are becoming more excitable. So if they are depolarizing, they are becoming more excitable, then you have more action potentials, you have more contractions or motility. Then you also have cholecystokinin. Cholecystokinin is also stimulatory to the smooth muscle cells. It can increase motility in the small intestines. But remember, the same cholecystokinin was inhibiting motility and secretions in the stomach, but here in the small intestines, it's going to facilitate motility. Then we also have a hormone which is called motilin, of which I told you to say motilin is produced when you're starving. So even when the stomach is empty, there is some sort of motility there that is facilitated by motilin hormone. And this motilin hormone is going to enhance motility by causing depolarization of the interstitial cells of Kegel. The frequency of firing slow waves in spike potentials, they're going to increase. And you know to say that will translate into an increase in the motility of the small intestines. There are also some inhibitory hormones that are produced by the GIT and also other organs. So secretin, it's inhibitory to segmental contractions. So secretin is going to reduce the contractions, and you know to say secretin is produced by the S cells in the upper portion of the small intestines. So the upper portion of the small intestines, you have the duodenum and part of the ileum that can produce some secretin. So you have those S cells, which are special modified mucosal cells that are responsible for production of secretin. Of course, for these hormones to be produced, there is need for a stimuli. So there are different types of stimuli that are stimulating the cells to produce the hormones, of which we also looked at hormonal control, and you know the stimuli that is stimulating these cells. Even if you don't understand now, there is a lecture that is dedicated for secretions. So we'll also look at mechanism by which these hormones are secreted. Glucagon, on the other hand, it's inhibitory to segmental contractions. Okay, so that is all on control of segmental contractions. So in the duodenum and proximal jejunum, you find that the frequency of these segmental contractions or slow waves is 12 slow waves per minute. So even the frequency of segmental contractions is to be somewhere around 12 segmental contractions per minute. Then in the terminal ileum, the frequency is going to reduce because the activity of the interstitial cells of Kegel, they are also reducing. So you're going to have a frequency of 8 to 9 segmental contractions per minute because that's the frequency for the slow waves. So if each slow wave is resulting into a spike potential, that will stimulate the circular muscles to contract, the longitudinal muscles also to contract. Then, of course, the effect of autonomic nervous system, especially the vagus nerve, because we say the majority of the small intestine is supplied by the vagus nerve. So the parasympathetic stimulation via the vagus nerve, that can cause an increase in the motility. So the segmental contractions are going to increase because of parasympathetic stimulation via the vagus nerve. Why? We said acetylcholine as a neurotransmitter is stimulatory in function. So it's going to stimulate the smooth muscle cells to contract. On the other hand, the sympathetic nerve, they are going to reduce the movements. So they are inhibitory. Why? It's because they are the postsynaptic membrane or the postsynaptic neuron is going to release the norepinephrine, and the norepinephrine has got the opposite effect as compared to acetylcholine. The norepinephrine or epinephrine is going to inhibit the motility in the small intestines. So the segmental contractions also, they are going to be reduced by sympathetic nerve stimulation. Okay. What are the functions of segmental contractions? So just like we saw the general functions of motility, so they are similar here. What will be the functions of segmental contractions? These are called mixing contractions to some extent. Agitation of intestinal contents. So agitation of intestinal contact, because you're talking of chopping of chyme. So you are agitating the chyme to some extent so that you are mixing the chyme with digestive secretions, the enzymes, and also exposing the finished monomers that have been digested for absorption. So it tends to increase the degree of subdivision of food particles. You are chopping the food particles, which is called chyme. At this point in time, it's not very good to refer to it as food because now it has been converted into chyme. It's no longer food. So it's called chyme. So you can call it chyme or the ingesta. That's much better rather than calling it food. Because even if we remove it here, we give it to a person, they're not going to eat it. So it's no longer food at this point. It's called chyme. So the mixing of chyme with intestinal secretions. So mixing contractions. Then changing the layers of food in contact with the mucosa, facilitating absorption. So as you are having these segmental contractions, you are also changing the layers of the chyme in contact with the mucosa. That is going to facilitate absorption. So we are done with segmental contractions. We move on to pendular movements. The pendular movements, I know most of you know the pendulum and you know how it moves, the pendulum. A long time ago, we had wall watch, the watch that would put on the wall. So it's called a wall watch. Some of them, they had a pendulum. So that pendulum, you know how it moves. It swayes side by side. So you have those swaying movements side by side, okay? So even the small intestines, they can also have such kind of movements. So these are side to side swaying movements accompanied by lengthening and shortening of the intestine. So there's lengthening and shortening of the intestine that will bring about the swaying movements. I'll explain the actual mechanism. So what is the function? The function are similar to segmentation contractions. So you're talking of agitation of the chyme, mixing of the chyme, changing the layers of the chyme in contact with the absorptive surfaces. So all those functions are similar here. So as you're having those swaying movements, you are also mixing the chyme, you are also changing the layers of the chyme in contact with the absorptive surfaces, you're also mixing the chyme with the digestive secretions. So it's, in terms of function, it's similar to that of segmental contractions. So if a question comes to say, can you explain the functions of pendular movements in the small intestine? You're not just going to say the functions are similar to that of segmental contractions. You need to explain them, okay? So this diagram here is just summarizing the mechanism of pendular movements. So the swaying movements side by side. So you can see. So what is going to facilitate the side by side swaying movement of the intestine? It's because of unilateral contraction of both circular muscles and longitudinal muscles. So unilateral, if you look at the intestine here, you have the left part and the right part. So if I'm saying unilateral contraction, it means that one side, on the one side of the intestinal smooth muscle cells are only contracting, the other side are not contracting. So for instance, if on this side, the smooth muscle cells are stimulated to contract, on the other side, they are not contracting. So the contraction of this side, it will cause this segment to shorten. So as the segment is shortening, it will sway to the other side. Then the other side now will contract so that the other side relaxes. I'm talking about, let me just use right and left for you to understand. So let's say this is a loop of intestines that is involved in pendular movement or swaying movements. So on the left side of the intestine, the circular muscles and the smooth muscles are contracting. On the right side are not contracting. So it will sway on this side. Then the opposite will happen. The left side, they will relax, the right side will contract, and then it will sway on the other side. So you have these swaying movements of the intestine, okay? If you don't understand, when we have a lab, I think I'll explain on this one, the swaying movements, so that I demonstrate how it happens. But just not to say it's caused by unilateral contraction of both circular muscles and longitudinal muscles. So both circular muscles and longitudinal muscles are contracting, but it's not everywhere. It's unilateral contraction that will bring about swaying movements. Okay. So we proceed, we discuss peristalsis. So the peristaltic contraction, the mechanism is very interesting, so you need to pay attention and understand. So peristaltic contractions or peristalsis is defined as a wave of contraction preceded by a wave of relaxation that travels arborally. So the movement is from the oral cavity towards the anus or away from the oral cavity. That's why it's called arboral or arborary travel of the chyme. So you have a wave of contraction that is preceded by a wave of relaxation. So this wave of contraction preceded by a wave of relaxation is due to the neurotransmitters that are released by the intrinsic nervous supply. So there are local reflexes that are involved in peristaltic contractions that will bring about smooth muscle cells to contract behind the bolus and smooth muscle cell on top and beyond the bolus to relax. So that mechanism I'll explain. There's a diagram for me to explain that. So you can see here that you have peristaltic contraction in the small intestines. You have this wave of contraction that is preceded by a wave of relaxation. So the smooth muscle cells behind the bolus, they are stimulated to contract. Then the smooth muscle cells above and beyond the bolus, they are inhibited, so they are going to relax. So the only way this bolus is going to move is to move downwards. Why? Because behind it, there's a contraction that is squeezing the bolus to move to the segment that are relaxing. Okay, so this mechanism is well coordinated, and I'll explain how it is coordinated. So peristaltic contraction, so you can see the movement of the bolus. So they are more like propulsive movement that will propel the bolus to move from point A to point B. So there is a wave of contraction behind the bolus and a wave of relaxation above and beyond the bolus. So these cells will be inhibited to contract. They will relax to increase the lumen of the GIT. Then also behind you have contraction that is reducing the lumen of the GIT, pushing the bolus to move forward. So this movement, it's arboreal, meaning that it's moving away from the oral cavity towards the anus. So it has to be maintained. But you need to know that sometimes you can have reversed peristalsis, especially when you're about to vomit. You can have reverse peristaltic movement, especially in the upper small intestine, in the duodenum. So some of the duodenal content can also be pushed back into the stomach. So it's possible for a person to vomit chyme that is coming from the small intestine, or that is coming from the intestine. Why? It's because sometimes because of pathology, toxins, you can have reversed peristalsis that will now move things from the anal cavity towards the anus, which is not the normal peristaltic contractions. The normal peristaltic contractions, the direction has to be arboreal, okay, or anulwards if you want, towards the anus. The control, you can't run away from the nerves when you're talking of control of peristaltic contractions. So it is neurogenic and depends on the myenteric plexus, especially the myenteric plexus. Remember, you have the myenteric and the submucosa. But when you're talking of motility, we're interested in the myenteric plexus, the one that is between the longitudinal and the secular muscles. So they move anulward. So the movement is anulward or arboreal at the velocity of 0.5 to 2 centimeters per second. That's the velocity of how the bolus is going to be moving. So after 1 second, it will cover a distance of 0.5 to 2 centimeters. So they are very weak and they'll die out at a distance of 3 to 5 centimeters. So each peristaltic contraction that has been initiated, it will die off at a distance of 3 to 5 centimeters. It's not going to proceed. You need to initiate more peristaltic contractions at that point again for the bolus to move. Otherwise, they are so weak, they will die off when they cover a distance of 3 to 4 centimeters. So there is a net movement of 1 centimeter per minute. So this is the net movement of the chyme or the bolus. So the bolus will be propelled to move at least 1 centimeter per minute due to peristaltic contractions. So they are starting, they will die off, others will start, they'll die off just like that. Then the bolus or the chyme will propel forward. So the control of peristalsis, okay? The contraction wave is conducted only in arboreal direction. So these are the starring and the Baylis law. So these are just like laws that are governing peristalsis, or that will try to explain peristalsis. So what you need to understand here is that the movement is just towards the anus. So it's arboro direction. Then it requires the integrity of my enteric plexus because it's the one that is controlling the GIT. Okay. So in this diagram now, I'm including the neurotransmitters that are involved in the connection. So in the mucosa of the GIT, there are sensory receptors. Those sensory receptors, some of them, they are responding to stretching. So they are called mechanosensitive receptors or stretch receptors. They are going to be stimulated once there is stretching there or distension of the mucosa by the presence of the chyme that is moving or the bolus. So as the chyme is moving in the lumen here, it's going to stimulate the stretch receptors. So the stretching by the foot, this stretching is going to stimulate the stretch receptors that are connected to the sensory nerve. So some of these receptors are free nerve endings, others are modified receptor cell that can generate a receptor. Okay, so this slide is just explaining the mechanism of peristalsis, of which I've already started. I've told you to say peristaltic contractions. It will require the intact degree or integrity of the enteric plexus because it's the one that is involved in control of peristaltic contractions. It will involve local reflexes. So because they are reflexes, peristaltic contractions are more of reflexes, they need a stimulus. So this stimulus is going to be stimulating the sensory free nerve endings. So you have sensory receptors that are associated with the mucosa. They are sensitive to stretching or they are sensitive to mechanical pressure. So they are called mechanosensitive channels or mechanosensitive receptors. So as the chyme is passing in the lumen of the small intestines, that chyme is going to be distending the lumen, hence stimulating the stretch receptors. The stretch receptors are connected with the sensory nerve fiber, which is called the afferent nerve fiber. So the afferent nerve fiber is projecting to the enteric plexus. So it's going to project to the enteric plexus to stimulate the enteric plexus. So you can see the interneuron of the enteric plexus is being stimulated by the sensory fiber here after stimulation by stretching. So the stretching means that there is chyme that is moving there, then you need to increase the motility. So what happens next? So the interneurons that are being stimulated here via a stimulatory neurotransmitter, in most cases is serotonin. So serotonin is the one that is being released here, and also acetylcholine. So acetylcholine is going to stimulate these interneurons, and the interneurons, they are communicating with other interneurons, or they are communicating with the motor neurons that are innervating the smooth muscle cells. So the next neurons, the final neurons here, are motor neurons that are innervating the actual smooth muscle cells. So those that are projecting forward, they are going to release nitric oxide, VIP, which is vasoactive intestinal peptide, and also ATP. These are the neurotransmitters that will bring about inhibition of motility. So the smooth muscle cells are going to be inhibited by hyperpolarizing them. Then once the smooth muscle cells are hyperpolarized, they become less excitable. So if they are less excitable, there will be relaxation. So the smooth muscle cells above and beyond the bolus, they are going to relax because of the neurotransmitters that are being released there. Nitric oxide, VIP, vasoactive intestinal peptide, and ATP, adenosine triphosphate. So these are inhibitory. Then the neurons behind the bolus that are projecting behind the bolus, they are going to release neurotransmitters, acetylcholine and substance P. So the acetylcholine and substance P, they are stimulatory in function. So they are going to stimulate the smooth muscle cells. So the smooth muscle cells are going to contract behind the bolus. So once they are contracting, the lumen of the GIT is going to decrease, or it's going to reduce because the circular muscles are contracting. So that now will propel the bolus to move to the next point, because the next point, there is relaxation taking place. So the movement is arboro direction. So this is a movement of peristaltic contractions, a borrow direction, okay? But it might get a bit complicated, the same peristaltic mechanism, because if it comes as an essay question, I would want you to be very smart and very specific about the information. So in that case, I would want you to draw something like this. So you need to be able to draw, label, and explain the diagram. So I'll explain what is happening here. So I've already told you to say the stimulus is stretching. That is done by the movement of the bolus. So this is the movement of the bolus. So as it was passing here, it was stretching. So the stretch sensors are going to be stimulated. So the stretch center that are stimulated here by the previous passage of the bolus is going to stimulate an action potential. So the stretch receptors will depolarize, then they will initiate an action potential that will be propagated via the sensory nerve. So the one shown in green here is a sensory nerve that is communicating with the stretch receptors. So it's going to propagate or transmit an action potential towards the interneurons. So we have two interneurons here. There's one shown in blue and the other one in brown. So this is brown or purple. Yeah, this is brown and blue. So these are interneurons. So the interneurons, they can either communicate with the motor neurons that are innervating the muscle fibers. So you can see the interneuron shown in blue here is communicating with the motor neuron. The motor neuron is the one shown in brown, dark brown here. So the motor neuron is innervating the smooth muscle cells. So you can see some branches of the motor neuron is innervating the longitudinal muscles above the bolus. Then it's also sending another branch to innervate the secular muscles before the bolus or behind the bolus. So once this motor neuron has been stimulated by the interneuron, it's going to send action potentials or transmit action potentials that will stimulate the longitudinal muscles above the bolus to contract. So the longitudinal muscles above the bolus, they're going to contract. When these longitudinal muscles are contracting, the segment or the GIT is going to shorten because the longitudinal muscles are contracting. Remember the orientation of longitudinal muscles and also secular muscles? Then the secular muscles behind the bolus, they are going to be stimulated to contract as well. So you can see the contraction of the secular muscles that will decrease the lumen, pushing the bolus forward. Then you have the other interneuron that is shown in yellow. This yellow interneuron is communicating with the motor neuron shown in red here. That is innervating the longitudinal muscles above and beyond the bolus. It's inhibitory. So there are inhibitory neurotransmitters that are released here, like nitric oxide. Then you have VIP, vasoactive intestinal peptide. Then you also have ATP. So that is going to inhibit these secular muscles to contract. So there is relaxation. So the lumen is going to increase, then the bolus can easily move. Then it's also communicating with other interneurons. So you have the interneuron that is shown here. I think this is purple now. This purple interneuron is communicating with the motor neuron shown in red. So this motor neuron shown in red, it's inhibitory. So it's also releasing inhibitory neurotransmitters. So these secular muscles that had contracted, they will be inhibited so that they can now relax. So there will be disinhibition that is taking place there. So the disinhibition will result into the secular muscles to relax because the bolus has passed this segment. So this is how peristaltic mechanism looks like. So down here, there's a list of the neurons, whether they are stimulatory or inhibitory. So the ones with a plus, it means they are stimulatory. So they are going to release stimulatory neurotransmitters. The one with a minus, they are inhibitory. So it means they're going to be releasing inhibitory neurotransmitters, okay? And the type of the neurotransmitters. So you have serotonin, you have acetylcholine, then you have VIP, which is vasoactive intestinal peptide. So in simple terms, let me just explain. I've told you to say that peristaltic contractions, you have the sensory neuron that is communicating with the interneurons. So this sensory neuron, once it fires an action potential, is going to stimulate the interneurons. So the interneuron that is innervating the longitudinal muscles above the bolus, they are going to be stimulated to contract. So it means here there is release of acetylcholine in substance P, because we say those are the neurotransmitters that will bring about muscle contraction. And also the circular muscles behind the bolus, they are going to be stimulated to contract. So the longitudinal muscles, when they are contracting, the segment is going to shorten. When the circular muscles are contracting, the lumen is going to decrease. So this bolus will be pushed. But the circular muscles above and beyond the bolus, they are going to be inhibited. There is release of nitric oxide, VIP, vasoactive intestinal peptide, and adenosine triphosphate, ATP. So this is the actual mechanism. Okay, so in this short video, under 1 minute, it's just illustrating the same peristaltic contractions. Okay, so you see it here. I don't know whether you are seeing what I'm projecting. So I'm interested in the intestines. So you can see those peristaltic contractions, but here they are taking place within the large intestines, which are also referred to as mass movement of substances that haven't been digested in the production of the feces or fecal material. But you can see that there's a wave of contraction that is preceded by a wave of relaxation, of which the mechanism for peristaltic contractions I've already explained. So at your own time, you can watch the illustration just to jerk your memory to remember the peristaltic contractions. But you know, I was just explaining from the diagrams. I always add some notes for you to understand. So just a bit of notes on the peristalsis mechanism. So it appears that local stretch releases serotonin, which activates sensory neurons that activate the meningeal plexus. So the meningeal plexus is going to be activated. The cholinergic neurons that are passing in the retrograde direction in this plexus activate neurons that release substance P and acetylcholine, causing smooth muscle contraction. So those that are running in the retrograde, meaning that they are running behind the bolus, they're the ones that are releasing substance P and acetylcholine to cause smooth muscle contraction. And then at the same time, the cholinergic neurons passing in an anterograde direction, they are going to activate neurons that secrete nitric oxide, VIP, and ATP. The nitric oxide, vasoactive peptide, and ATP, they are going to produce relaxation ahead of the stimulus. And this stimulus is stretching, which is a bolus or chyme that is being propelled in the lumen or the GIT. Factors that influences peristalsis. So what factors can enhance or inhibit peristaltic contractions? So it is increased after a meal. You know, to say after a meal, there's a lot of distension on the GIT. So once the GIT is distended more, even the peristaltic contractions are going to increase. So of course, after a meal, you expect that peristaltic contractions are going to increase. So this is caused by gastroenteric reflexes. And on top of that, you know, to say that you have the intrinsic and the extrinsic nervous supply to the GIT. So the gastroenteric reflexes, these are local reflexes that will be initiated by the presence of chyme in the GIT. Then you have hormones that can enhance and those that can inhibit. So here, just like segmental contractions, you have gastrin, cholecystokinin, inulin, and serotonin. These are going to enhance the motility of GIT. So peristaltic contractions is going to increase. Secretin, glucagon is also going to inhibit peristaltic contractions, just like we say that the segmental contractions are also inhibited by secretin and glucagon. So it's the same thing here. So if you got it at first, even here you get it right. Even if a question comes, you'll be able to understand because it's the same information. It will apply for peristaltic contractions and also segmental contractions. Then the function of peristaltic contractions, progression of chyme towards the ileocecal valve. So it will propel the chyme towards the ileocecal valve so that you have now emptying of the ilium into the cecum. The movement of chyme from the ilium into the cecum via the ileocecal valve. Then they are also involved in spreading of the chyme along the intestine. That will also facilitate absorption and digestion. Then sometimes you can have peristaltic rash that can result into diarrhea. So these peristaltic rash are simply powerful irritation of intestinal mucosa. So if you have powerful irritation of intestinal mucosa, it's going to stimulate strong peristaltic contractions, so that whatever is contained within the lumen or the GIT that is irritating the GIT, it has to be removed from the GIT. So you have peristaltic contractions so that whatever is in there can easily move to the colon, and then you can defecate in the form of a diarrhea. So that will function to relieve the small intestines from the irritating chyme. So you need to relieve yourself from these irritants so you can have peristaltic rash that is also coordinated. So it will be initiated by physical or chemical irritation. So whatever is irritating the GIT, that can also trigger peristaltic rash. So you have powerful and rapid peristalsis that will sweep the contents into the colon. It can be caused, this could be the cause of infective diarrhea. So in infective diarrhea, if you have infection, certain infections can cause diarrhea. Why? It's because when there's infection, it could be that infection, there are microbes that are producing toxins that are irritating the lumen of the GIT or the mucosa of the GIT. So once the mucosa of the GIT is irritated, it can trigger peristaltic rash, so that whatever toxins are there are going to be propelled towards the colon, then you'll be able to defecate. Okay, but it will be some kind of a diarrhea, so that you get rid of the irritants that are causing irritation. Movement of the villi. So the villi can also move. Those finger-like projections associated with the mucosa, they also have special types of movements. So they are initiated by local nervous reflexes that occur in response to chyme. So chyme, again, is acting as a stimulus to cause local reflexes that will produce movement of the villi. The muscularis mucosa extends into each villus as a filament of the smooth muscle fiber. So these are the ones that are going to initiate the villi to contract. Okay. So what sort of movements can you appreciate by the villi? So we have lashing movements and the villus pump. Lashing movement and the villus pump. So what is the function of these movements? These movements are going to accelerate the flow of blood. So they will have an effect on the flow of blood that is involved in absorption, and also lymph, absorption of fat, because we have the lacteals. So the movement of lymph is going to be enhanced by the movement of the villi. So the lashing movements and the pumping movement. Then it's also going to increase absorption. So you know to say these movements or the villi, they're going to increase absorption because the blood flow is also going to increase. So here is the diagram that is showing the two types of movement. So the first one is the villus pump. That is due to the shortening and lengthening of the villi. So when you have those muscularis mucosa contracting, the villi is going to shorten. If you have relaxation, the villus is going to relax. So you have shortening and lengthening of the villus. So these are the finger-like projections. Then you have lashing movements. So if you have unilateral contraction of those muscularis mucosa, then you are going to have side-by-side movements. So these are called lashing movement. Okay, so they move like wipers of a car. You know those wipers of the car, the way they move. So those are lashing movements. So even the villi, they can actually exhibit those kind of movements, which are called lashing movements. Okay, so that's it with the motility in the small intestines. So we're not going to end here. We still have a bit of time. I'm going to share another lecture so that we also discuss motility in the large intestine. Okay, so I can only hope that you are seeing what I'm projecting. I'll put every mic on mute so that you don't disturb my class. So we proceed. Like I said, if you have a question, just raise your hand. I'll be able to see. Then I'll unmute you then you'll be able to speak. Okay, so we continue. So we've already started looking at motility in the intestines. We've discussed motility in the small intestines. So we are moving on to the large intestines. So we'll discuss the emptying of the ileum, just like we discussed the emptying of the stomach, gastric emptying. So here we're going to discuss now the emptying of the ileum, because now the chyme has to move from the small intestines to the large intestines so that you are able to appreciate the motility in the large intestines. So the motility in the colon simply summarizes the motility in the large intestines. So these motility, the mechanisms are similar. So you already know how peristaltic contractions come about. Those are mixing contractions. But here in the large intestines, they will pick different types of names. So we will talk of mass movement in the large intestines. Those are simply peristaltic contractions in the large intestines. The mechanism is similar. So I'll be a bit fast so that we cover this lecture in 30 minutes, starting now. Okay, so we have the ileocecal valve. The ileocecal, it's a sphincter. So in most cases, it's referred to as a valve because it's going to protrude into the cecum. So it looks like a valve that will prevent the backflow of chyme into the ileum. So hence the name ileocecal valve as opposed to a sphincter. But it operates like a valve. It also operates like a sphincter because there are smooth muscle cells there that can contract to close off the opening of the ileocecal valve. Okay, so you can see it in this diagram here. What is contributing to the ileocecal valve? So there are circular muscles of the cecum. So you can see here, there are these circular muscles of the cecum that is contributing to the ileocecal valve. Then you also have the longitudinal muscles of the cecum. So the longitudinal muscles of the cecum, so some of them, they are projecting to the ileocecal valve. Then you also have longitudinal muscles of the cecum that are continuous with the longitudinal muscles of the ileum. So this part is the ileum, this part is the cecum. Then you can see the ileocecal valve that is protruding into the cecum lumen. So you have these longitudinal muscles and the circular muscles that will form the valve. So when these smooth muscles are contracting, the valve is going to close. When they are relaxing, it's going to open. So the emptying of the chyme is regulated because you don't want to dump all the chyme from the ileum into the cecum. So there is some sort of control that will allow movement now of the chyme from the ileum into the cecum. So you can see the contributory smooth muscle cells. So you have the circular muscles and longitudinal muscles of both the cecum and the ileum that are contributing to the formation of the ileocecal valve. Okay, so this is more of anatomy. We're interested in the physiology. So emptying of the ileum is the first thing because chyme has to move from the ileum into the cecum. So emptying of the ileum, the ileocecal sphincter, also known as the ileocecal valve, separates the terminal end of the ileum from the cecum, the first part of the colon. So what is separating the two is the ileocecal valve. It's found between the ileum and the cecum. So the ileocecal valve itself protrudes into the lumen of the cecum and it's closed when excess pressure builds up in the cecum. So as the ileum is emptying its contents into the cecum, there is buildup of chyme in the cecum that will create pressure, and that pressure will collapse the valve. So it will cause the closing of the valve as well. And on top of the contraction of the circular muscles and the longitudinal muscles that are part of the ileocecal valve. So normally this sphincter is closed. In most cases it's closed, but it can be regulated to open to allow emptying of the ileum. Distension of the distal ileum promotes peristalsis in the ileum and the opening of ileocecal sphincter. So when you have peristaltic contractions in the terminal part or the ileum or the distal ileum, that will promote the contraction of the ileum. And as the ileum is contracting, is squeezing the chyme that will be now emptied into the cecum. That is a process of ileocecal emptying or the ileum emptying into the cecum. So distension of the cecum causes the ileocecal sphincter to close. So when a lot of chyme has been emptied into the cecum, it will cause distension of the cecum that will cause the closing of the ileocecal sphincter or ileocecal valve. So it will close. The resistance to emptying at the ileocecal valve prolongs the stay of chyme in the ileum. That will facilitate absorption. So that resistance of ileocecal valve opening, it will facilitate absorption to take place because now chyme will spend much time in the ileum. That will provide enough time for absorption to take place. So in this diagram, you can see the portions of the large intestines, starting with the cecum. The cecum, so this is where you find the ileocecal valve. You can see this protrusion into the cecum. This is called the ileocecal valve. That is connecting the ileum and the cecum. Then the first part of the colon is the ascending colon. Then you have the transverse colon, the descending colon, the sigmoid colon. Then you have the rectum and the anus. So this is the motility that we are appreciating in this segment of the intestines, the large intestines. So motility in the colon. So the colon serves as a reservoir for the residues of meals that cannot be digested or absorbed. So any substances that haven't been digested or absorbed, it will be stored within the large intestine. That's why I'm saying that the colon or the large intestine will serve as a reservoir for the residues of meals that cannot be digested or absorbed. So it's involved in the production of the feces, so fecal material production. Motility in this segment is likewise slowed to allow colon to absorb water and some of the electrolytes like sodium and other minerals. So it doesn't mean that in the large intestine, there's no digestion taking place there, there's no absorption. There's a minimum of digestion taking place there because we can have some microflora or bacteria that are involved in digestion in the large intestines. There are a bit of enzymes there also that are involved in digestion, but the large intestine mainly is also involved in the absorption of water. So absorption of water is taking place in the large intestines. Even in the other parts of small intestines, there is still absorption of water, but the remaining water within the chyme, it will be absorbed by the large intestines. So that movement is slowed down. The motility of the colon is slowed down to allow absorption of water or the fluids and also electrolytes. So later on, we'll discuss digestion and absorption. So you know what sort of absorption and digestion is taking place also in the large intestine. So by removal of about 90% of the fluid, it converts the 500 to 2000 mils of isotonic chyme that enters each day from the ileum to about 200 to 250 mils of semi-solid feces. So here, this sentence is just telling you to say that the large intestine is responsible for reabsorption 90% of water in the chyme. So if 90% of water in the chyme is being reabsorbed or it's being absorbed by the large intestine, then that chyme is being converted from a fluid or from more like a solution into a semi-solid feces. So now you have the production of semi-solid feces. Why? It's because most of the water is being absorbed by the colon. Okay, 90% of water is being absorbed. Then we also have propulsive movements, which are called mass movements. So these mass movements are the same peristaltic contractions that are now taking place within the colon or the large intestine. The large intestine is innervated by the parasympathetics. So the first part of the large intestine, which is the ascending and some portion of the transverse colon, is heavily innervated by the vagus nerve. So the vagus nerve is part of the parasympathetics that is innervating this part of the large intestine to modulate or to regulate peristaltic contractions and other sort of motility that is taking place in the colon. But the distal part of the large intestine, especially the descending, the sigmoid, the rectum, and also the anal sphincter, they are heavily innervated by the pelvic nerves. Remember, I told you to say the sacral segment of the spinal cord, especially S2, S3, S4, is contributing to the spinal nerves that will innervate the distal part of the large intestine. So the distal part of the large intestine is heavily innervated by the pelvic nerves that are coming from S2, 3, and 4 segment of the sacral regions. So you need this part to be heavily innervated by the pelvic nerves because this is a part that is involved in the defecation reflex. So the defecation centers are found in the sacral region of the spinal cord. So even if you have a spinal cord injury above the sacral region, the defecation reflex is not going to be compromised. So you have patients with spinal cord injuries, but you know to say the defecation reflex is still intact, even if they have a paralyzed maybe leg or whatever it is. So you need to understand the clinical implication of that, especially the innervation. So you need this innervation to be very good because this is a part that is involved in the defecation reflex. So you need a lot of parasympathetics that can stimulate the distal part of the large intestine for it to be involved in the defecation reflex because the muscles need to contract. Motility in the colon, so we can divide the colon into certain segments. So let's start by looking at the motility of the cecum in the proximal colon. So the motility of the cecum in the proximal colon is the cecum in the proximal part of the ascending colon. Okay, so what sort of motility can you appreciate there? So most contractions of the large intestine are segmental. So you have segmental contractions, which are mixing contractions. So you need to have these mixing contractions that will facilitate absorption. Because remember, as you're having these mixing contractions, they are also changing the layer of chyme in contact with the absorptive surfaces so that you can absorb as much water as possible, as much minerals as possible. So you have absorption of electrolytes and minerals that is taking place within the large intestine. That's why you have also a lot of segmental